Hello, everybody. My name is Mahshad Hashemi. I'm a master's student in artificial intelligence under supervision of Dr. Mansoori in Shiraz University. Today, I want to present our work titled Identifying Novel Disease Genes Based on Protein Complexes and Other Protein Features. First, I will briefly introduce the problem. After that, some related works will be introduced. And in the end, our methods and datasets will be discussed. At first, let me start with a brief biology introduction, which will be useful in the process. Humans are multicellular beings that have cell nuclei. In every cell of a healthy human, 23 pairs of chromosomes exist, and they are made out of twisted strings of DNA. 95% of our DNA is called non-coding DNA, which means we don't know their functions yet, and only 5% of our DNA has functional role. There is an important concept in biology called central dogma, which basically shows how proteins are produced out of genes in DNA. After proteins are produced, they link together and protein complexes are made. Protein complexes are the foundation of many biological processes. They perform a variety of biological functions. Diseases typically have three origins: environmental factors, microorganisms like viruses and bacteria, and genetic factors. Down syndrome, hemophilia, and CF are some examples of genetic diseases. There are some diseases that have some multi-factorial origins, such as heart disease, diabetes, and Alzheimer's disease. As you can see, huge portion of diseases have genetic related factors, and identifying these factors is needed to find a cure or produce effective drugs. Genotype and phenotype are two expressions that are widely used in this work. The genotype is a set of genes in DNA responsible for unique traits, and phenotypes are the visible or expressed traits. For example, in these examples, DNA is the genotype, thumbprint and height are phenotypes. In this part, I want to introduce some of the papers that we used in our work. In the first paper, Kohler and others utilized two datasets, AMIM and PPI, and they introduced a method called RWR, or random walk with restart, to prioritize candidate disease genes. They put candidate disease genes with known disease genes on a PPI network, and then used known disease genes as seed nodes in the algorithm. And in the end, they prioritized candidate disease genes based on their adjacency to the known disease genes. For better understanding, let's have a look at random walk and random walk with restart algorithms. Random walk gives us the closeness of two nodes in the graph, which in that M is the probability of a movement between different nodes in a graph. RWR algorithm is the same as a random walk, but with a slight difference, which gives the walker the ability to jump to any randomly selected nodes in the graph. And as a result, it avoids walker to be trapped in a dead end. In the next paper, Wu and others introduced a method called a cipher, which integrates the PPI network and phenotype information obtained from AMIN. In this algorithm, a regression model has been built. At first, it will calculate the similarity between every phenotypes, and then it calculate the adjacency of every candidate disease genes to each phenotype. And in the end, based on the closeness of genes to phenotypes and the closeness of phenotypes to query phenotype, it will produce a degree for each candidate disease gene, and with that, it will prioritize candidate disease genes. Uh, in the next paper, Lee and Patra introduced a new method called RWRH. They integrated protein network with phenotype network based on gene interactions and built a heterogeneous network, as you can see in the picture. In this network, phenotypes and genes are ranked simultaneously. They use this method to understand the correlation between different diseases. In other words, for a single disease, they use phenotypes and genes as seed nodes, and other diseases are ranked based on them. In the next paper, Yang and others used three datasets, AMIM, Chrome, and PPI, and built a three-layered a network with those. They introduced a method called RWPCN, which basically is the same as RWR, but extended in a way that can be used on protein complexes. In this algorithm, each node is a protein complex, and they try to prioritize candidate disease genes based on protein complexes. In the next paper, Divan and others tried to find the disease genes by using a weighted PPI network, which had genes as nodes and used a string dataset for the weights, which contains protein-protein interaction values. Then they used a Dijkstra algorithm on the network, which prioritized the candidate disease genes. Okay, I'm going to introduce our datasets now. Three datasets were used in our work. The first dataset is HPRD, which contains human proteins and their interaction values. It contains about 10,000 proteins related to humans, and there are like 41,000 interactions between them. The second data set, is called Coral, which contains about 4,000 protein complexes related to humans. And the last dataset is called OMIM, which contains about 17,000 gene phenotype relationships related to 1,000 disease phenotypes. Okay, and now I want to explain the overall structure of our heterogeneous network. Our network has three separate layers. In the first layer, we have a phenotypic network which contains the disease phenotypes and their interactions. In the second layer, we have protein complex networks, which are extracted from Chrome dataset. These two networks are connected with the gene phenotypes information that are available in OMI dataset. And in the last layer, we have PPI network. We use proteins as nodes and their interactions as edges. And as mentioned before, protein complexes are made out of several proteins, and by using Chrome dataset, we can extract their interactions. Uh, in next part, we want to see how these networks are constructed. The first network that I'm going to explain is the protein complex network. For building a protein complex network, we should find several neighbors for each complex based on their functional similarity. We calculated their similarity, or better to say, their closeness based on the interactions between proteins involved in those complexes. As you can see in the pictures, Each complex contains several proteins, and they might have some interactions. For example, P1, P2, P3 belong to complex A, and P4, P5 belong to complex B. Um, P1 and P3 from complex A, P5 and P4 from complex B are connected together. The degree of closeness between two complexes is higher if we can find more connected proteins. Um, Okay, if I want to formulate my explanation, it will be something like that. In this formula, E is the closeness degree between complexes CA and CB, and VCA is the set of nodes that are involved in complex CA. Um, the formula basically counts all the interactions and divides it by the multiplication of all unique nodes of CA and CAB. For constructing the phenotypic layer, again, we need to find some neighbors for each phenotype. Based on previous works, we chose 10 as the number of neighbors for each phenotype. For a better comprehension, I'm going to explain this matter with an example. Imagine we have three phenotypes with their related genes and complexes. The first one in orange is our query phenotype. As you can see, both gene 1 and gene 3 belong to complex A, and gene 2 belongs to complex B. Now we want to find 10 neighbors for our query phenotype. If the genes that are related to our query phenotype and the genes that are related to the phenotype which we want to, to see if it's suitable to be considered as a neighbor belong to the same complex, we choose that phenotype as a neighbor with the highest degree, which is 1. If we couldn't fill all 10 spaces with phenotypes with these attributes, we should choose from phenotypes that are related to complexes in the neighborhood of the query complex, and we give them the degree of E, which we calculated before. And this is the formula. Pheno score is the degree between two neighbors in the phenotypic layer. In this part, we want to see our algorithm. I want to mention again that we aim to prioritize candidate disease genes based on known disease genes. And as mentioned before, each query phenotype will have 10 neighbors, and for each neighbor, we have a score called a phenoscore. Each of these neighbors have several genes, and all of these genes together are called seed genes. So every member of this set, like S, has a score that is calculated with this formula. First, we should see how many phenotypes are related to this specific gene, and then calculate the summation of related phenoscores and divide it by the summation of all phenoscores. Uh, in the second part of our algorithm, we want to find initial scores for protein complexes based on their seed genes. So we multiply the density, the formula is provided, of that complex to the summation of all seed scores related to that complex. Okay, now it's time to use RWR on our protein complex network. M here is the transition matrix of protein complex network. P0 is the initial probability, and we set R to 0.8. and then we run our algorithm. When the difference between pt plus 1 and pt becomes lower than 10 powers minus 10, we consider that a steady stage has been reached and stop the iterative process. In this stage, every protein complex has a final score, and the last part of algorithm, which is calculating the scores for all of the genes begin. Gene score equals to the summation of the final scores of all complexes that contains the current gene. After this process, we will prioritize all of the genes based on their gene score and select the top 20. Okay, now I want to explain my metrics. We used a leave one out cross validation to compare our methods with others. Each gene phenotype interaction is a test sample. P is our query phenotype, and G is its related disease gene. In each iteration, we will remove the interaction between P and G. After running our algorithm, genes will be prioritized based on their connections to the query complex. If the test gene is ranked as first, then we consider it as a successful prediction, else it's a failed prediction. We used two methods. First, whole genome evaluation, and second, ab initio evaluation. In whole genome evaluation, we only omit one of the interactions between query phenotype and its related genes. If the gene that we omitted its interaction with the query ranked first, it's a successful prediction, else it's a failed prediction. In ab initio evaluation, we omit all of interactions between the query phenotype and its related genes. We only use the genes related to query phenotype neighbors as seed genes. Again, if one of the test genes ranked first, it's a successful prediction. Now it's time to see our results. We compared our work with four other methods by utilizing a whole genome evaluation and ab initio evaluation. As you can see, our work outperforms the previous methods in both sections. And we also chose two diseases, type 2 diabetes and breast cancer, as our case studies. In here, we can observe the obtained result by choosing breast cancer as our query phenotype. Among the top 20 genes that we selected, we had six confirmed genes, 11 suspicious genes, and three unmatched genes. Um, and as you can see, our unmatched genes in both case studies are reduced, and our suspicious genes are increased, which is a good result for a case study. For our second case study, we chose the type 2 diabetes as our query phenotype. Again, we selected the top 20 genes, and among them we had 8 confirmed genes, 7 suspicious genes, and 5 unmatched genes, which once again reduced the number of unmatched genes and increased the number of suspicious genes. That's it. Thank you all for your attention.